Hi, good morning. First of all, I would like to thank the organizing committee for the nice invitation. So it's a pleasure to be here on the second Lisbon International Conference on Social Science Humanity. So I'm going to share my screen. So I'm from Lisbon. So my institute is a high school engineering from Lisbon, where we have 11 bachelor's degrees and master's degrees. So you are all invited to visit our webpage, and if you want to visit, please contact us. So today I'm going to speak a little about my work concerning biomarkers discovery based on FTIR spectroscopy. So what is a biomarker? Is not obligatory. A molecule or a set of molecules could be a fingerprint, could be a process, something that we could quantify it and associate it to a phenotype as a diagnostic, a prognostic, or monitoring the efficacy of a therapy. So today, worldwide, they are applying these kind of techniques for severe disease as cancer. And as an example of the National Institute of Health in the United States, they try to apply this kind of biomarkers discovery process for a precision medicine for 1 million persons. So together genetic information, the phenotypic characteristic of that person to discover new biomarkers. So an example of the relevance of biomarkers in cancer is breast cancer, where we already know some biomarkers that if you know these characteristics of the breast cancer will be going to define a better and more precise therapy. Like for instance, if the cancers overexpress estrogen or progesterone receptors, mostly the most efficient therapy will be based on endocrine therapy. In opposition, if the overexpression receptors is another different receptor as human epidermal growth factor, so we could apply a completely different therapy, as for example, based on monoclonal antibody directed for this kind of receptors. So this is already known, these biomarkers for breast cancer, but also for other kinds of cancers. So, and these biomarkers will define, of course, a better diagnosis, a more precise diagnosis, and a more precise therapy. So, and how to discover these biomarkers? Well, the more efficient process is based on system biology, where the goal is to evaluate key elements like DNA, RNA, proteins, and metabolism in interaction on a defined systems. For that, use another kind of technologies like the omics science, that we want to evaluate as many as possible these molecules in the systems, like the genome. And in the last 20 years, there's a lot of evolution of this kind of techniques. For instance, more or less 20 years ago, the first human genome that was sequenced, it took around 10 years and more or less $3 billion, and there was a multi-international institutions involved. So involved thousands of people worldwide, $3 billion, more than 10 years. And nowadays, after and most of the evolution of the last 10 years on the techniques, in this case based on next sequence generation, we are able to sequence a whole human genome on a couple of days for a couple of $1,000. So it's much cheaper, much more rapid. So this evolution was also achieved in other kind of techniques, not looking for the genotype, but focus also on the phenotype. So if you, instead of looking what the cell can do, you could look at what really the cell is doing, looking at proteins and even more on the metabolites. So the phenotype. For this, there are several kind of techniques. Neither of them are perfect. All of them are looking for a set of this type of molecules, just looking for instance, on the chromatography techniques associated to mass spectroscopy for detection of the metabolites. All of these techniques are looking for a different set of metabolism. There's no perfect solution. So more problematic is the complexity of the process. Usually we need high complex processing of the sample to acquire these metabolites, derivatize the samples, some of the process, and after we have a very complex data analysis, as because we are generating a lot of data, and the variability of the human population is so high that it turns the highly complex this data analysis. So as an alternative, we could look for more simple techniques like as Fourier transform infrared spectroscopy. Why? Well, this technique is very much simpler to apply. Sometimes the processing of the sample is minimal as a dehydration step. It's very rapid to acquire a spectrum, 1-2 minutes for a given sample. And there are very alternative methods of detection. So let's see some examples. For instance, on this one, we apply the technique on bacteria culture, Escherichia coli, recombinant diverse kind of strains. And we developed very good regression models with correlation coefficients almost near 1, that is, and very low error associated, and from a single spectra acquired in real time, and in each a couple of minutes we retrieve a lot of information, as for instance the concentration of acetate, ethanol, glucose, cholesterol, and even the plasmids inside the cell. So it's a very precise technique. We also applied, that one was conducted in the near infrared with fiber optical case, so giving in real-time data. This one was applied on animal cell cultures, human mesenchymal stem cells and expansion procedures. And here we also achieved very nice regression models, and we applied in the mid-infrared spectra. So the technique is highly versatile, work on near infrared or mid infrared with in real-time acquisition or high throughput methods, for instance, based on multiplates with microwells, as using 5 to 20 microliters of samples. So it's very versatile. However, that needs calibrations, and calibrations takes long and could be also highly complex. So in alternative, you could look directly for the spectra without these calibrations. So, and for that, we could apply the convolution method, as usually we apply to chromatography, now for spectroscopy. So applying, for instance, the second derivative, we could resolve superimposed peaks like this one and retrieve information along the culture of the process, as for instance the content of the energetic status of the cell along the time, apparent transcription levels, and so on. So we could retrieve the metabolic profile of the culture along the time in a very simple mode. So I'm going to give you 2 examples for searching biomarkers after showing that the sensitivity and the versatility of the technique, as for instance, discovering genotoxicity biomarkers. On this case, we collaborate with another group, and that group applied a human trial. We consider 92 human volunteers. Half of them were hospital professionals that unfortunately were exposed to genotoxic drugs, as observed after the surface of most of the places where they work were contaminated by that drugs. And the other half of the participants were not exposed to this kind of genotoxic substances. So the other team analyzed it by conventional methods, the genotoxic potential of this exposition by acquiring peripheral blood, extracting the lymphocytes of this blood, and conducted a cytokinase blocking assay to observe after the nucleus of these lymphocytes. So after the optical observation of the nucleus and the abnormalities of the nucleus as micronucleus, they observed that the exposed population presented in average a statistical significance higher number of these abnormalities. So the second step we conducted was to evaluate it if in average and using the spectra, we also observe significant differences between the exposed and not exposed population. So again, based on the second derivative spectra for results superimposed spectra, we observe in diverse regions of the spectra that in average, the exposed population presented on these particular regions statistical different values than to non-exposed population as these wave numbers as an example. And the next step, we also observed, considering the whole spectra to the mid-infrared region, that the principal component analysis of the second derivative spectra, that most of the exposed population formed a cluster on the score plot in different regions, and this output was also observed in the hierarchical clustering analysis that is on the right. So most of the exposed samples were in a different cluster in relation to exposed sample. We were even possible to develop classification methods based on supervised modeling as support vector machines, where from a single spectra we're able to predict if that person was or not exposed to this genotoxic substance. In other words, if they present genotoxic values as compared to the conventional method, and were possible with high sensitivity, precision, and accuracy. And even the area under the curve was very high, always near a 0.8, 0.99. But in this new methodology, the test is much simpler to conduct, much more rapid, a couple of hours. The quantity to sample, the procedure to sample is much simpler because we are based on a very drop of blood. On the second and last example, we applied a completely different technique based on Fourier transform infrared spectroscopy. On this case, we evaluated tissues from biopsies from prostate cancer. So we evaluated using a focal plane array. That's this kind of so it's a microscope associated with a focal plane array, where our array has 64 for 64 detectors. So for a final region, we are acquiring 4,096 spectrum, and for each tissue, we acquired 10 images. So we are having hyperspectral data that is very difficult to manage in computational power. So just to show an example, this is a microscopic observation of prostate. This is adenocarcinoma, and this is hyperplasia of the prostate. From the infrared region of the spectra, we integrate some areas. For instance, this is completely proteins, we have here lipids, and integrating these regions, we were able to reconstruct based on infrared spectra the image that we were observing at optical microscopy. So this was a simple exercise, but our main goal was to achieve a diagnostic. So for that, we analyzed the average spectra of the hyperplasia versus the adenocarcinoma. And in average, they are different. However, the standard deviation are huge, as expected, because we are looking based on human samples and a tissue with a high diversity of characteristics of cells, capillaries, the glands, and so on. So for that as conducted in previous examples, that was not manageable to conduct the diagnosis. So, and the second step, we analyzed the combination of several pre-processing methods, including several degrees of derivatives, We also analyze diverse non-supervised and supervised models. The non-supervised models didn't work to separate the data, in contrary to the previous observation, and the supervised models worked. So we would shift by one of these kind of supervised models an accuracy of diagnostics of around 97% that is used. Is that one considering the classification by the pathologist that is based on human observation of the microscopic optical images. That is a very complex procedure and laborious, time-consuming, and it has a human error associated because it's based on the human experience. For that, in our case, we have automatic method, much more quick. So you could say that this microscopic observation of tissues at hyperspectroscopy, because we are using focal plane array detectors, is one of the most rich and concise way to represent the role of the omics, because we are not just looking for the metabolome or the proteome, because we analyze at once all these kind of molecules as the metabolites, proteins, and even nucleic acids. So in resume, this kind of techniques presents very appealing characteristics for biomarkers discovery, as is very simple to apply, could be conducted in high throughput or tissue analysis by image. It's much more economic, captures the whole molecular fingerprint in a very high sensitive specific mode, so is very appealing. So I thank you very much for your attention. This is my contact. If you want to collaborate, it would be a pleasure to you. So thank you again to the organizing committee for the nice invitation, and hope to see you soon, presential in Lisbon in the following conference. So thank you.